Hello, folks. Hoping that you are all doing well and ready to learn something new with us. So, today we are going to discuss all about emulsion. This is what we will discuss. 1. What is emulsion? 2. Types of emulsion. 3. Tests for identification of emulsion type. 4. Formulation of emulsions. 5. Classification of emulsifying agents. 6. Other emulsion formulation additives. 7. Preservation of emulsions. 8. Physical stability of emulsions. 9. Chemical instability of emulsions. 10. Stability testing of emulsions. 11. Manufacture of emulsions. Let's begin. What is emulsion? An emulsion is mixture of two liquids that would not normally mix. That is to say, a mixture of two immiscible liquids. By definition, an emulsion contains tiny particles of one liquid suspended in another. Chemically, they are colloids, where both phases are liquids. They are typically milky in appearance, and the suspended material may be colloidal in nature. The globule size of emulsion ranges between 0.25 to 25 micrometers. The sizes of globules influence the quality of emulsion. The larger globules form coarse emulsions, normal globules of mean size, 5 micrometers, form fine emulsions, and smaller globules of size, 10 nanometers, forms microemulsions. Next are types of emulsion. Pharmaceutical emulsions usually consist of mixture of an aqueous phase with various oils and/or waxes. Let's learn the different types of emulsion. Oil and water emulsion. If the oil droplets are dispersed, that is, dispersed phase, throughout the aqueous phase, that is, continuous phase, the emulsion is termed as oil and water emulsion. For example, oral emulsion, parenteral nutrition, vanishing cream. Water and oil emulsion. A system in which the water, that is, dispersed phase is dispersed throughout the oil, that is, continuous phase is termed as water and oil emulsion. For example, cold cream. Here is the diagram for comparison of these two types of emulsion. It is also possible to form multiple emulsions. For example, many small water droplets can be enclosed within larger oil droplets, which are themselves then dispersed in water. This gives a water and water emulsion. The alternative oil and water emulsion is also possible. Upcoming are tests for identification of emulsion type. Several simple methods are available for distinguishing between oil and water and oil emulsion. The most common of these involve miscibility tests with oil or water. The emulsion will only be miscible with liquids that are miscible with its continuous phase. Conductivity measurements. Systems with aqueous continuous phases will readily conduct electricity, whereas systems with oily continuous phases will not. Staining tests. Either a water-soluble or an oil-soluble dye is blended with the emulsion. Microscopic emulsion will show whether the continuous phase or the dispersed droplets are colored. Forthcoming is formulation of emulsions. Because of the very wide range of emulsifying agents available, considerable experience is required to choose the best emulgent system for a particular product. Let's discuss it in detail. First is choice of emulsion type. For oral administration, fats or oils for oral administration, either as medicaments, in their own right, or as vehicles for oil-soluble drugs, are invariably formulated as oil and water emulsions. In this form, they are pleasant to take, and the inclusion of a suitable flavor in the aqueous phase will mask any unpleasant taste. For intravenous administration, they must also be the oil and water type, although intramuscular injections can also be formulated as water and oil products if a water-soluble drug is required for depot therapy. Emulsions are most widely used for external application. Semi-solid emulsions are termed as creams, and more fluid preparations are called either lotions or, if intended for massage, into the skin, liniments. Both oil and water and oil types are available. Water and oil emulsions will have an occlusive effect by inhibiting the evaporation of eccrine secretions. This will hydrate the upper layers of the stratum corneum, and so is particularly useful for dry skin conditions. Second is choice of oil phase. In many instances, the oil phase of an emulsion is the active agent, and therefore, its concentration in the product is predetermined. Liquid paraffin, castor oil, cod liver oil, and arachis oil are all examples of medicaments which are formulated as emulsions for oral administration. Cottonseed oil, soybean oil, and safflower oil are used for their high calorific value in emulsions for intravenous feeding. Examples of externally applied oils that are formulated as emulsions include turpentine oil and benzyl benzoate. Many emulsions for external use contain oils that are present as carriers for the active agent. It must be realized that the type of oil used may also have an effect on both the viscosity of the product and the transport of the drug into the skin. One of the most widely used oils for this preparation is liquid paraffin. This is one of a series of hydrocarbons that also induces hard paraffin, soft paraffin, and light liquid paraffin. They can be used individually or in combination with each other to control emulsion consistency. The film-forming capabilities of the emulsion can be further modified by the inclusion of various waxes, such as beeswax, carnauba wax, or higher fatty alcohols. Third is emulsion consistency. The texture or feel of a product intended for external use must also be considered. A water and oil preparation will have a greasy texture and often exhibits a higher apparent viscosity than oil and water emulsions. This fact is often used to convey a feeling of richness to many cosmetic formulations. Oil and water emulsions will, however, feel less greasy or sticky on application to the skin, will be absorbed more readily because of their lower oil content, and can be more easily washed from the skin surface. Emulsions of high apparent viscosity for external use are termed creams and are of semi-solid consistency. They are usually packed into collapsible plastic or aluminum tubes, although large volumes or very high viscosity products are often packed into glass or plastic jars. Next is choice of emulsifying agent. The choice of emulsion to be used will depend not only on its emulsifying ability, but also on its route of administration and, consequently, on its toxicity. Although there is no approved list of emulsifying agents for use in pharmaceutical products, there is an approved list of emulsifiers as food additives for use in the European Union. It can be assumed that emulsifiers contained in this list would be suitable for internally used pharmaceutical emulsions. The regulations mainly include naturally occurring materials and their semi-synthetic derivatives, such as polysaccharides, as well as glycerol esters, cellulose ethers, sorbitan esters, and polysorbates. It will be noted that Most of these are non-ionic. These have a tendency to be less irritant and less toxic than their anionic, and particularly their cationic, counterparts. The concentrations of ionic emulsifying agents, necessary for emulsification, will be irritant to the gastrointestinal tract and have a laxative effect. Thus, they should not be used for oral emulsions. When choosing an emulgent for parenteral use, it must be realized that only certain types of non-ionic material are suitable. These include lecithin, polysorbate 80, methyl cellulose, gelatinant, serum albumin. Adjacent is formulation by the HLB method. First, it is need to understand that what is HLB value, the hydrophile-lipophile balance, or HLB of a surfactant, is a measure of the degree to which it is hydrophilic or lipophilic. It is a value between 0 to 60 defining the affinity of a surfactant for water or oil. HLB numbers are calculated for non-ionic surfactants, and these surfactants have numbers ranging from 0 to 20. Physically stable emulsions are best achieved by the presence of a condensed layer of emulgent, as the oil-water interface, and the complex interfacial films, formed by a blend of an oil-soluble emulsifying agent with a water-soluble one, produce the most satisfactory emulsions. A useful method has been derived for calculating the relative quantities of these emulgents necessary to produce the most physically stable emulsion for a particular oil-water combination. This is called the hydrophile-lipophile balance, or HLB method. Following that is use of phase inversion temperature. The use of the HLB system has several disadvantages, including the inability to take into account the effects of temperature, the presence of additives, and the concentration of emulsifier. It is possible to overcome some of these problems. Oil and water emulsions, stabilized with non-ionic emulsifiers, change to water and oil emulsions as the temperature rises, when the hydrophilic and lipophilic properties of the mixed emulsifier are just balanced. The phase inversion temperature, or PIT, is the temperature at which the emulgent has equal hydrophilic and hydrophobic tendencies, and the emulsion will invert. Now, we will learn the classification of emulsifying agents. The inclusion of emulsifying agent, or agents, is necessary to facilitate actual emulsification during manufacture and also to ensure emulsion stability during the shelf life of product. There are many types of emulgent available, but for convenience, they can be divided into two main classifications. Synthetic or semi-synthetic surface active agents. Naturally occurring materials and their derivatives. Synthetic and semi-synthetic surface active agents. There are four main categories of these materials, depending on their ionization: in aqueous solution, anionic, cationic, non-ionic, and amphoteric. Let's discuss them in detail. Anionic surfactants. In aqueous solutions, these compounds dissociate to form negatively charged anions that are responsible for their emulsifying ability. They are widely used because of their cheapness, but because of their toxicity, they are only used for externally applied preparations. Alkali metals and ammonium soaps. Emulgents in this group consist mainly of the sodium, potassium, or ammonium salts of long-chain fatty acids, such as sodium stearate. Soaps of divalent and trivalent metals. Only the calcium salts are commonly used. A number of amines form salts with fatty acids. One of the most important of these is based on triethanolamine, and it is widely used in both pharmaceutical and cosmetic products. Sulfated and sulfonated compounds. An example is the sodium lauryl sulfate or SLS. It is used with C-tostierol alcohol to produce emulsifying wax, which stabilizes such preparations as aqueous cream and benzoyl benzoate application. Sulfonated compounds are much less widely used as emulsions. Materials of this class include sodium diactylsulfosuccinate and are more often used as wetting agents or for then detergency. The most important group of cationic emulsions consists of the quaternary ammonium compounds. Because of the toxicity of cationic surfactants, they tend to be used only for the formulation of antiseptic creams, where the cationic nature of the emulgent is also responsible for the product's antiseptic properties. Cetramide, that is, cetyl trimethylammonium bromide, is one of the most useful of these cationic emulsions, and is used at a concentration of 0.5% with 5% C-tostierol alcohol for the formulation of cetramide cream. Non-ionic surfactants. Non-ionic emulsions are particularly useful because of their low toxicity and irritancy. Some can therefore be used for orally and parenterally administered preparations. They also have a greater degree of compatibility with other materials than do anionic or cationic emulsions, and are less sensitive to changes in pH or to the addition of electrolytes. Amphoteric, or zwitterionic surfactants possess both positive and negative ions. According to the anion type, amphoteric surfactants may be classified into lecithin, butene, imidazolene, and amino acid type. Amphoteric surfactants have minimal toxicity. They are moderate to the skin and possesses excellent biodegradability. They are mainly used in the development and manufacture of suspensions used as topical lotions and hair shampoos. They are not for internal medicines. Naturally occurring materials often suffer from two main disadvantages. They show considerable batch-to-batch variation in composition and hence in emulsifying properties, and many of them are susceptible to bacterial or mold growth. For these reasons, they are not widely used in manufactured products requiring a long shelf life, but rather for extemporaneously prepared emulsions designed for use within a few days of manufacture. Naturally occurring materials and their derivatives include natural polysaccharides. The most important emulsifying agent in this group is acacia. Semisynthetic polysaccharides. Several grades of methylcellulose and carmellose sodium are available and exert their action in a similar way to that of acacia. Sterol-containing substances, beeswax, wool fat, and wool alcohols are all used in the formulation of emulsions. Beeswax is used mainly in cosmetics creams of both oil in water and water in oil type. Beeswax is used as a stabilizer for water in oil creams. Other formulation additives include buffers, density modifiers such as sucrose, dextrose, etc., humectants such as glycerol, polyethylene glycol, antioxidants such as butylated hydroxyanisole or BHA, flavors, colors, and perfumes, sweetening agents such as sucrose, sorbitol, etc. It is essential that a suitable preservative be included as part of an emulsion formulation, particularly if naturally occurring materials are to be used. This is to prevent the growth of microorganisms that may be present in the raw material and or introduced into the product during use. Some of the natural products, particularly if they are to be applied to broken skin, should be sterilized before use. Bentonite, for example, may contain Clostridium tetani, but can be sterilized by heating the dry powder at 150 degrees centigrade for 1 hour or by autoclaving aqueous dispersions. Because of the complex systems involved, and the many factors to be taken into consideration, it is necessary to test the efficiency of a new preservative in the finished product and container by suitable, challenge testing procedures. A suitable emulsion is one in which the dispersed globules retain their initial character and remain uniformly distributed throughout the continuous phase. Several mechanisms cause emulsion breakdown. Let's learn them in succession, along with their prevention. Creaming is separation of emulsion into two regions, one of which is richer in the disperse phase than the other. An everyday example is the creaming of milk, when fat globules slowly rise to the top of the product. This is not a serious instability problem, as a uniform dispersion can be reobtained simply by shaking the emulsion. Creaming is, however, undesirable pharmaceutically because of the increased likelihood of coalescence of the droplets, owing to their close proximity to each other. The rate of the creaming can be reduced by these methods. Production of an emulsion of small droplet size. Increase in the viscosity of the continuous phase. Reduction in the density difference between the two phases. Control of disperse phase continuation. Flocculation involves the aggregation of the dispersed globules into loose clusters within the emulsion. The individual droplets retain their identities, but each cluster behaves physically as a single unit. Flocculation is the secondary minimum, occurs readily, and cannot be avoided. Redispersion can easily be achieved by shaking. Primary minimum flocculation, however, is more serious, and redispersion is not so easy. The presence of a high charge density on the dispersed droplets will ensure the presence of a high energy barrier, and thus reduce the incidence of flocculation in the primary minimum. The coalescence of oil globules in an oil and water emulsion is resisted by the presence of a mechanically strong, absorbed layer of emulsifier around each globule. This is achieved by the presence of either a condensed, mixed monolayer of lipophilic and hydrophilic emulgents, or a multimolecular film of a hydrophilic material. Hydration of either of these types of film will hinder the drainage of water from between adjacent globules, which is necessary prior to coalescence. As two globules approach each other, their close proximity causes their adjacent surfaces to flatten. As a change from a sphere to any other shape results in an increase in surface area, and hence in total surface free energy, this globule distortion will be resisted, and drainage of the film of continuous phase from between the two globules will be delayed. The presence of long, cohesive hydrocarbons chains projecting into the oil phase will prevent coalescence in a water and oil emulsion. Although it is not possible to list every incompatibility, these general points will illustrate the more common chemical problems that can cause the coalescence of an emulsion. Many of the oils and fats used in the emulsion formulation are of animal or vegetable origin and can be susceptible to oxidation by atmospheric oxygen or by the action of microorganisms. The resulted rancidity is manifested by the formation of degradation products of unpleasant odor and taste. These problems can also occur with certain emulsifying agents, such as wool fat or wool alcohols. Oxidation of microbiological origin is controlled by the use of antimicrobial preservatives, and atmospheric oxidation, by the use of reducing agents or, more usually, antioxidants. The contamination of emulsions by microorganisms can adversely affect the physiochemical properties of the product, causing such problems as gas production, color and odor changes, hydrolysis of fats and oils, pH change in the aqueous phase, and breaking of the emulsion. It is therefore necessary to include an antimicrobial agent to prevent the growth of any microorganisms that might contaminate the product. Adverse storage conditions may also cause emulsion instability. It has already been explained that an increase in temperature will cause an increase in the rate of creaming, owing to a fall in apparent viscosity of the continuous phase. At the other extreme, freezing of the aqueous phase will produce ice crystals that may exert unusual pressures on the dispersed globules and their adsorbed layer of emulgent. Methods of accessing instability include microscopic examination, globule size analysis, viscosity changes, accelerated stability tests, storage at adverse temperatures, centrifugation, and rheological assessment. It has already been explained that the smaller the globules of the disperse phase, the stronger will be the rate of creaming in an emulsion. The size of these globules can also affect the viscosity of the product. In general, it has been found that the best emulsions with respect to physical stability and texture possess a mean globule diameter of between 0.5 and 2.5 micrometers. The choice of suitable equipment for the emulsification process depends mainly on the intensity of shearing required to produce this optimum particle size. Other considerations, however, include the volume and viscosity of the emulsion and the interfacial tension between the oil and the water. The presence of surfactants, which will reduce interfacial tension, will aid the process of emulsification, as well as promoting emulsion stability. The advantages of emulsions as pharmaceutical products include: pharmaceutical emulsions may be used to deliver drugs that are poorly soluble in water, but readily soluble in oils. For example, in oil and water emulsions, the drug substance is dissolved in the discontinuous or internal oil phase. Following oral administration, the oil droplets, and hence the drug, may then be absorbed, using the normal absorption mechanism for oils. Some drugs are more readily absorbed when administered as an emulsion than as other, oral comparator formulations. Pharmaceutical emulsions may be used to mask the bitter taste and odor of drugs in which the drug is dissolved in the internal phase of an oil and water emulsion. The external phase may then be formulated to contain the appropriate sweetening and flavoring agents. Drugs that are more stable in an oily phase compared to an aqueous medium can show improved stability in an emulsion dosage form. Intravenous emulsions of contrast media have been developed to assist in diagnosis. Emulsion can be used to prolong the release of the drug, especially semi-solid emulsions, thereby providing sustained release action. The oily phase can serve as a reservoir of the drug, which slowly partitions into the aqueous phase for absorption. Essential nutrients like carbohydrates, fats, and vitamins can all be emulsified and can be administered to bedridden patients as sterile intravenous emulsions. Pharmaceutical emulsions may be employed to administer drugs to patients who have difficulty swallowing solid dosage forms. Emulsions provide protection to drugs which are susceptible to oxidation or hydrolysis. Emulsions are used widely to formulate externally used products, like lotions, creams, liniments, etc. Pharmaceutical emulsions are thermodynamically unstable, and therefore must be formulated to stabilize the emulsion from separation of the two phases. This is, by no means, straightforward. Pharmaceutical emulsions may be difficult to manufacture. Storage conditions may affect stability. They are bulky, difficult to transport, and prone to container breakages. They are liable to microbial contamination, which can lead to cracking. Uniform and accurate dose may not be achieved. In the pharmaceutical industry, they are used to make medicines more palatable, to improve effectiveness by controlling dosage of active ingredients, and to provide improved aesthetics for topical drugs such as ointments. Non-ionic emulsions are most popular due to their low toxicity, ability to be injected directly into the body, and compatibility with many drug ingredients. Cationic emulsions are also used in certain products due to their antimicrobial properties. Here, we reach at the finish line. Please subscribe our channel if you haven't subscribed it yet. We hope that these videos are helpful and informative for you. We welcome your suggestions and feedback. Until then, take care.